Thank you for joining us today and welcome to today's webinar. My name is Priya and I'm your host for today. And if you're attending a Firefly event for the first time, welcome. If you have been here before, good to see you again. As always, good to see you coming back for more information around Firefly's performance, applications, and so on. A couple of housekeeping pointers to make sure everything goes smoothly and you get the best out of today's session. Here's a screenshot. Here's a screenshot of your attendee interface. You should see something that looks like this on your own computer desktop in the upper right-hand corner. You're listening in using your computer's speaker system by default. If you would prefer to join over the phone, just select telephone in your audio pane and the dial-in information will be displayed. The duration of the presentation is about 40 minutes, followed by a Q&A session. This is your opportunity to ask the presenters your questions. You can submit them at any time during the presentation by typing them into the questions chat box of your control panel. We'll then gather them all for our speakers to address at the end. Without further ado, let me introduce Paul Lomax, who is our product manager for genomics here at SPT LabTech. Today, Paul will give you an overview of Firefly, the next-gen liquid handler and its key features. I'm also really pleased to introduce Dr. Anita Pearson, who leads the applications development for Firefly. Anita is an experienced scientist, having worked for 15 years developing methods and instruments for next-generation sequencing in the industry. Today, Anita brings to you hot-off-the-press data generated from Firefly. And with that, let me hand over to our first speaker, Paul, to kickstart the webinar. Over to you, Paul.
Thank you, Priya. I'm going to give you a short introduction to the Firefly, just an overview of the system and its features, and a little touch on its applications, and also some video content just to give you an idea of what it looks like in operation. And then I'll be handing over to Anita, who will be actually giving the main presentation around a specific application for library prep. So let me move into the first point about the system. If I can just get my slides to advance, there we go. So here, firstly, the dimensions of the system. So it's quite a compact unit. It's just 780 millimeters high, 560 millimeters deep, and 660 millimeters wide. So it's a very compact unit with an awful lot of functions built into this small system. It's all run from a touch screen that is mounted at the front of the system, so you don't lose any further bench space to an extra PC. What you see there is all you need. And then there are two kind of sides to the system. So on the left-hand side, you've got a 384 pipetting head that can be used for 384, 96, or columns of 8 or 16 tips for 96 or 384 well plates. And on the right-hand side, you have a choice of either 3 or 6 non-contact dispensing heads, which are great for dispensing things like master mix and beads and so on. And then on the left-hand pipetting head, there is also a gripper, so you're able to move things between the decks on the system, and I'll talk about those in a moment. And you can also move plates between things like magnets, so on and off magnets, onto shaker blocks, onto heater cooler plates, and so on. So really giving you all the functionality that you need for a wide range of applications. There are two decks on the system, and this is why we've got such a compact system. So we use the vertical space. So one deck sits over the other deck, so you've got an upper and a lower deck. Each of those have got 8 SBS positions, so for 8 microplates or things of the same footprint. And then we also utilize the space below these two decks. So on the left-hand side, we have a shaker and a plate heater cooler position. And on the right-hand side, we also have the option for a heater cooler for the reservoirs for the dispense system. So essentially those 3 or 6 syringes on the dispense system can aspirate from reservoirs that are held below the 2 operational decks. So to give you a little overview of the pipetting capability, the pipetting head is a standard air displacement system. So it is a 384 tip head. It can load either 384 tips, 96 tips, or strips of 8 or 16. So it's very flexible and adaptable, working between either 96 or 384 well plates, and also either whole plates or partial plates. So it's very adaptable. The tips are auto load, so the tips come in cassettes of 96 or 384 or in specific strips, and these load into a holder that goes onto the deck for then automatic tip changing, so the tips can be changed as required during a protocol. And the range of volumes goes from 0.5 microliters up to 125 microliters, which is a little reduced if you're using filter tips. We're also developing what we call a 4-in-1 adapter, which expands the volume to a higher range up to 500 microliters, but that would just be for the 96 format and also the 8 strips, so that's not applicable to 384 watt plates. And then in terms of the dispensing heads, you've got the choice of 3 or 6, as I mentioned earlier. Each of these dispensers has an independent disposable syringe that has a volume range that's very wide, from 200 nanoliters all the way to 4 milliliters. So essentially it's a 4 milliliter reservoir for non-contact dispensing, and then you can dispense multiples of the same or variable volumes from that syringe. And each syringe is independent, so you can be dispensing different volumes, different wells with different liquids, or you can be multiplying and using multiple syringes for the same reagent to get a faster dispense. These are disposable, but generally they would be used throughout a protocol or even multiple protocols during the day if you're not changing the reagent that you're using in that tip. So you can really cut down on plastic consumption. If you compare that to using pipette tips to do the same operation, you can really reduce the amount of plastic that you dispose of. The really nice thing is that these dispense heads are positive displacement, and they're genuinely liquid class agnostic. So we can dispense a wide range of reagent types, from very viscous reagents and enzymatic reagents and so on, through to things like beads. And you don't really need to change the settings at all. You can just run with the standard settings and dispense with ease. They're also capable of working across the standard plate formats, even up to 1536 well plates. And it's also very fast, so we can dispense 5 microliters across the 384 well plate using 3 channels in less than a minute. And if we used all 6 channels, that would be less than 30 seconds. Here's just an outline of the range of tips that are available. So we've got a range of volumes. We do a smaller volume tip as well for greater accuracy down at the low end of the volume range. They're available in sterile and filter formats. And you can see here how they get loaded onto the system. So we have a common loading stand that the tips are loaded into, placed on the deck, and then the head picks them up as and when it needs them. If you're using the strip tips, there's just a comb adapter that goes into the head, and then the strips can be slid in into the head for use. We also have a wide range of application-ready accessories. So we've been using the Alpaca Magnum Flex 96 magnet for our applications in 96 well plates, and we can supply those with the systems. But we plan to accommodate a wide range of magnetic blocks. Essentially anything that works in an SBS format could potentially be used on the system. We've also incorporated a shaker unit, so we use the BioShake 300 ELM, and we also use the Q Instruments cold plate. So these are part of what we call our genomics options kit. So these are optional add-ons that can be bought with the system or added later. And then we have a wide range of accessories for the dispense. So we have standard reservoirs, obviously the disposable tips. We have low dead volume reservoirs. And we also have some options for both passive cooling and then also active cooling. So we have another cold plate heater cooler option for the dispense channels. We're also very proud of the software that we've developed for the system. So it's a very easy to use graphical user interface. It features enhanced security, so logins and audit trails for the different users that use the system, and multi-user levels, so you can have kind of users who simply just access protocols and run them, or kind of advanced users who would write new protocols and kind of access to the full range of the software. It also kind of features some intelligent programming features whereby it makes some intelligent assumptions based on what you're doing to actually make the programming easier. I could probably explain that better in a full software demo if anyone is interested. Obviously, just let us know and we can arrange that. The other nice thing is it's open license software, so you can actually author on any PC. You can install the software on your laptop or your desktop, and you can program protocols from where you like and then just send them to the system. And that can be done via the cloud or over the network. So there is a cloud-based sharing system. And with that, we can also manage the history and version management of protocols. So if multiple users are opening protocols and making changes, those are recorded and you can track that. So it really is a very easy to use software, but also a very powerful software. And just in terms of the cloud, we're really, this we think is a really important aspect of the system. So I mentioned the ability to author protocols anywhere, but really we see this as a collaboration tool, either within sites and within companies, or actually outside and between different researchers across the globe. You've got control over that, how much you share, how little you share, but we really hope that that's going to really start driving forward the ability for users of Firefly to share protocols, labware, definitions, liquid classes, and so on. As we ship the systems, there will be a number of protocols available. So one of the ones that's going to be discussed today is here, so the NebNext Ultra 2 FSDNA, but also the standard DNA and the RNA kits from NEB, and then also the Illumina DNA prep. And then there's a long list of applications that we'll be adding to that. So typically something like one per month will be added and available over the cloud to download. And then finally, I'll just leave you with just a quick video just to give you a sense of the Firefly in action. So let me just start this. So here it's just picking up a 384 head, and so it's picking up the disposable tips onto the 384 head. You can see that's coming from the lower deck, from the stand that the tips are placed on. The deck at the top now moves across, so we can go and aspirate samples from the 384 well plate. And then it's going to dispense that across to a second plate, and then it's going to move across to dispense using the dispense heads. And we could be doing this with a mixture of 96 and 384 tips, moving from say 96 to 384 well plates. We could be using columns as well, so it's very, very flexible. Here you see it's just going across and dispensing with the 6 heads of the dispense head across this 384 well plate. So this combination of dispensing and pipetting in this compact footprint is really kind of unique in liquid handling systems, and also combined with the shakers and the incubators, it provides a full range of tools for setting up a wide range of applications, and obviously with a with a focus on NGS library prep, it's very well suited to that. So at that point I will stop, and I will hand over to Anita, who's going to tell you a little bit more about a specific application for NebNext Ultra 2 for the FSDNA kit.
So thank you, Paul, and thank you, Priya, for the introduction. So I've been working on the development of the Firefly instruments and also the development of protocols to run on Firefly. So the key application area we've been looking at for Firefly is next generation sequencing library preparation. So this tends to be a long process, many pipetting steps, so it's a really good candidate for automation. So today I'm going to take you through some of the dispense performance data we've collected using Firefly. The main part of my talk will focus on the protocol development work we've been doing. And I'm going to share with you some of our initial run data. I'll finish up by taking you through a short example protocol of where we to show you how we can actually normalize samples quite simply using Firefly. So dispense performance. So dispense performance is really fundamental to any liquid handler. It provides the base on which all applications are subsequently built, and also underlies our ability to achieve good sample-sample uniformity. So pipetting performance. So this data here was collected using our 384 channel air displacement head. So that's the head I'm just circling here on the left of this picture. So this single head can be used with 96 or 384 tip arrays or columns of tips. So this data relates to the dispense performance of the 96 and 384 tip arrays. So we used buffer for this test. We dyed it with tartrazine. And what we did was we aspirated this reagent and then dispensed it to a quantification plate. And we measured the tartrazine absorbance of the fluid in the wells. And we compared this absorbance to a standard curve. In this way, we could determine the volume of the transfer. So that's the data that is being shown here in the measured volume column. Using this measured volume, we could calculate the trueness of the dispense. So the trueness is a measure of how close our measured volume is to our target volume. We can also look at the variation in this measured volume by calculating the coefficient of variation, the percent CV. So what we found was that the variation, so the across all of our transfers was low. It was less than 3%, even when we're looking at dispense volumes of in the 1 to 2 microliter range. So I forgot to mention that we're actually looking at three different repeats for each of the dispensers, which were performed at different volumes. So we're very pleased with this data. It looked good, and even in the low volume range. So the trueness of the dispensers was also good. Typically less than 6% for all of our dispensers that we looked at, and the majority of the dispensers were far below even that level. So they were on target. So the non-contact dispensing dispense performance was looked at as well. So this was using the head on the right, our positive displacement head. So we used a range of different reagents for this testing. All reagents were dyed with tartrazine again. So we looked at some buffer and also some other more challenging reagents, which we called enzyme buffer and bead buffer. These had high concentrations of either glycerol or PEG in them. So these reagents are more similar in composition to those that are typically used for next generation sequencing library preps. So in a similar way to as we did before, these dyed reagents were dispensed and we measured the tartrazine absorbance and related that dispense volume back to a standard curve. So we looked at a range of different volumes and we actually performed either 384 individual dispensers or 96 individual dispensers, just depending on the target volume we're looking at. And this way we determined the actual measured volume for each dispense. And then in the same way as before, we calculated the trueness, so how close this measured volume was to the target. And we looked at, and we could calculate also the variation in the dispense volume by calculating a percent CV. So this data shows that we had low variation in our dispenses. Our CVs are typically less than about 3 to 4%, even with our more challenging reagents like that enzyme buffer or bead buffer. And the dispensers are also on target, typically all less than 6%. And if you look at the individual results, mainly far below 6%. So this shows that data that we've collected for Firefly, it shows that the dispensers are both repeatable and on target from both these liquid handling heads. So I'm going to move on to talk about protocol development for Firefly. So this is the approach we've taken, quite a long approach. So to start with, when developing a protocol for Firefly, we start by looking or determining which head to use for each dispense. So we typically use the dispense head, so the positive displacement head, for all bulk additions of reagents. So by this I mean reagent additions where we're dispensing the same reagent to all wells of the plate. We use the pipetting head, so the air displacement 384 head, to do all plate transfers, any tip mixing, and also any transfers to a waste plate. When we're writing protocols, we try and use the shaker wherever possible to minimize tip consumption. So the shaker is on deck, and we want to minimize tips that we use in the protocols. The next thing we tend to do when developing protocols is we go for a reagent matching exercise. So the problem we have with next generation sequencing reagents is that they're quite expensive, there's not much volume available, and they're colorless. So for development purposes, this is not that favorable. So what we do is we match the liquid properties of our kitted reagents to dyed in-house reagents. And in this way, this gives us access to large quantities of reagent we can look at to really look in detail at these dispensers. We are actually able to see the reagent, which has huge advantages. And also, if we want to, we can quantify the dispense because we've got that dye in the reagent, so we're able to quantify it if we want to. So, so this is what we do. It's not a perfect sort of approach, but it is a really great tool for product development. Nothing beats using actual kitted reagents, but for development purposes, this is a really useful tool. So what we do then is we look at each step of the protocol and we analyze it in great detail. We look at each aspiration, we look at the aspiration speed, whether it's suitable, is there air being aspirated, and we look at each dispense. We are looking to achieve clean tips after a dispense. We're lucky in that our tips we use for Firefly are all transparent, so you can really see what's going on inside the tip, and we can make sure that our dispensers are executing how we want them to. So we also can look at contamination because we've got our dyed reagents, so we can see if there's any splashing of reagent. And also, the presence of a dye makes it easier to determine whether any mixing steps we're performing have been conducted thoroughly. So we test protocols using these match reagents, some expired kitted reagents, and only then do we move on to use functional kitted reagents. So I'd say this preliminary work is really thorough, and in doing this, we can minimize the unknowns before we're working with these more expensive kitted reagents. Right, so I'm going to move on to talk about some of the work we've been doing on a specific library prep protocol. For those who aren't so familiar with this process, these are the steps which we are trying to achieve when conducting a library prep. So we start with our genomic DNA, we fragment that DNA, and then we add adapters to the ends of those fragments. So the addition of these adapters enables our samples to be indexed or barcoded. So if we've got gDNA from different individuals, we can subsequently mix these samples together for sequencing, but still differentiate between the different individuals. What these modifications to the ends of these DNA fragments also enable the library to bind to the surface on which it will be sequenced. So it's really important that all of these steps of the library prep need to execute correctly in order to generate a library that can actually be successfully sequenced. So we've been looking at the NEB Next Ultra 2 FS DNA kit. So key features of this kit are that fragmentation step is enzymatic. It's not physical shearing. We're using enzymes to perform that fragmentation. So this fragmentation is not an isolated individual step. It's actually combined with another step in the protocol. It's not an additional step you need to perform. With this kit, we can use the NEB adapters. So these adapters have a hairpin loop structure with a uracil at the end. Yeah. And this uracil can be removed by the action of an enzyme mix called user. And this enables the library to subsequently undergo PCR. So a feature of this kit is this addition and incubation with this user enzyme. So it's not, this step isn't always present in other library protocols. So I wanted to make you aware of that. So when running our protocol, our initial aims were to stay true to our manual protocol, the NEB Next Ultra 2 FS DNA manual protocol, in terms of volumes and master mixes. And we wanted to perform all incubations greater than 20 degrees C off the depth. This was to avoid any evaporation issues causing problems with volumes. The whole process was conducted in PCR plates because we wanted to do all heating steps on a thermocycler. So in this way, we prepared 4 sub-protocols for this NEB Next Ultra 2 FS DNA library prep. So these are the 4 protocols to be run on Firefly. So between each protocol, we are able to refresh our dispense head syringes and also replenish any tips for the air displacement head as well. So this slide shows an outline of the first sub-protocol we prepared. It demonstrates that we can, or how we can use the pipetting heads and dispense head and process modules all together to execute this protocol. So we're using the dispense head for the bulk additions. So bulk additions of these like user, the adapter, enzyme mixes. So these are additions to all wells of the plate. I've indicated the time it takes to fill the whole plate with this reagent. So from the dispenser to the first well to the last well of that plate. So it's typically about a minute. If we're using a slightly larger volume, the time of this dispense increases a little bit. So I've used, the pipetting head was used for the plate transfer from the DNA input plate to the reaction plate. And we've used shaker mixing where possible to minimize tip consumption. And we've got a thermal incubation here on deck. So the last part of this protocol is the movement of the plate to the thermocycler for this user incubation. So this is the second sub-protocol we created. It's the adapted cleanup. So it is showing a cleanup, and these steps are really common for any cleanup used in NGS library preps. So again, we're using the dispense head and pipetting heads together with the process modules to execute this protocol. In terms of protocol development, we found that after the addition of beads to the reaction plate, the well volume is quite high. It's just over like 120 microliters. So, and we found that mixing of this, mixing on the shaker at this point does not give us a good mix. We needed to tip mix. So we developed a really nice thorough mix sequence. We aspirate and dispense about 75% of the well volume. We aspirate from the bottom of the well, dispense towards the top of the well, and we perform 50 mix cycles. This gives a really good thorough mix. It's been well tested. Another thing I wanted to point out about this sub-protocol is the dispense of ethanol. So after we've removed the supernatant to waste, we've got our bead pellets present in each well, slowly drying. So we want to, as quickly as possible, dispense ethanol to all wells of the plate. So we're dispensing a relatively large volume of ethanol, but we want to do this as quickly as possible. So we were able to achieve this by splitting the dispense into two. We performed, what we did was first dispense 20 microliters of ethanol to each well of the plate. And this was done really quickly, it takes about a minute. And then we went back and we topped up to achieve the final desired volume of 200 microliters per well. So in this way, we were able to minimize the drying time of the beads. One other thing to point out is after dispense of our elution buffer to the reaction plate, we found that it was better tip mix at this point. We did try shaker mixing. That was okay, but tip mixing gives us a better result. Just more even mix, which was successful in every single well. This is the third sub-protocol we prepared. So this is the PCR prep. So we've got the dispense of PCR master mix, a mix on a shaker, and then we're using the pipetting head to transfer in our indexes, and then another mix before sending the plate off to the thermocycler. So what I wanted to point out here is that we are able to successfully perform this index transfer from the foil sealed kitted index plate. So sometimes with automation, you get the index plate being picked up by the pipetting head. What we found was that if we sufficiently pierce the foil seals, we don't see any such problems. So we use a strip tube to push a strip tube into the foil seals of the index plate to pierce them and to really open out those holes. And in doing this, the holes are sufficiently wide that when we're using our pipetting head to aspirate from the index plate, we don't have any problems with the transfer. The plate's not picked up, and we don't need to hold that index plate down. So set up for our live run, we use the following conditions. We use human gDNA as our input, 100 nanograms input. There's lots of protocol options for this, the FS kit. So fragmentation time, we selected 30 minutes. And in terms of our adaptive cleanup, we didn't do any size selection, we just did a single bead addition, and 4 cycles of PCR as is recommended by NEB. So we prepared 1 column of samples in a 96 well PCR plate, and the magnet we used was an alpaca magnet. So here I'm showing you some of the fragment analyzer data we got after preparing libraries on Firefly. So I'm showing you traces for each of the individual samples, each of the 8 samples here on the left, and I've combined and overlaid them all for the figure on the right. So we're looking at fragment size along the x-axis and essentially the abundance of fragments on the y-axis. And to note here, all our fragment size distributions for all the samples, they overlay for all 8 samples. This indicates that we're getting consistent library size distribution for all these samples. And it does show that the yield from the library prep is also consistent across all samples. So this is the same data here on the left, and I've just shown the breakdown of the fragment analyzer output. We're also showing you the concentration from the fragment analyzer compared to the qPCR that we performed on each of these samples as well. So in terms of library yield, this is in line with what would be expected from this kit, using the DNA input we used and the number of cycles of PCR we used. So yeah, it's in line with what NEB suggests you should get. So this is good. Also, the fact that we were able to get some qPCR data, some output, confirms that we do have functional library molecules. So it shows that the gDNA was not only successfully fragmented, but the adapters and indexes were successfully added to the ends of these fragments. And so if we were going to further, we could actually generate data if these libraries were sequenced. So all in all, we are seeing good sample uniformity with Firefly. So for me, I could not help but notice a small peak here. I was hoping for just the main library peak to be shown in this trace, but we got a small peak at 55 base pairs. So I had to look at this closer. It intrigued me. So I looked at a manual prep as well. So this is the same data as I showed previously on the left. I looked at a manual prep. We still get this peak if I perform a manual prep. So, this is not an artifact of automating the protocol. We can remove this peak if we perform an additional PCR cleanup. So in this, see the peak has been removed here in this lower figure. I haven't showed you the control for this, but it does show the presence of a peak. If we do perform that additional bead cleanup, it goes away. So this is the approach NEB suggests. If we do have any additional peaks, perform additional cleanup. So that's the way of getting rid of it. We have tried using more stringent bead ratios for the PCR cleanup. That had the effect of just reducing the yield, but the peak was still present. And we had I ran the fragment analyzer using samples from different points in the protocol, and found that this is actually present after the adapter cleanup. So possibly modifying the adapter cleanup might might be able to get rid of this peak. Basically, this is a feature of the setup we've used, I believe. To do the adaptive concentration, we've been using the DNA input and that bead ratio we're using for the cleanup. I have to say that this is a small peak. It's small, and it should not affect our sequencing yield. So moving on, always keen to make improvements to protocols. So I was made aware of a slightly different version of the NEBNext FS protocol. It was used for some of the COVID work. So this protocol, it actually combines the user and PCR dispenses and incubation. So in doing this, we have to do one less dispense, one less transfer to the thermocycler. Great. So we tried that out. It worked very nicely. So this is, again, one column of samples that we prepared. And I've overlaid all the traces here. We're seeing that all our traces overlay. The library size distribution is very uniform across all samples. So again, we've got, using Firefly, we can generate libraries with good sample uniformity. That's very good. So I did want to share some initial data we've collected looking at well contamination. So this is the setup that was used. We were looking at 3 wells in pink, which received no DNA input, and we surrounded each of these with completely with samples where we had 100 nanograms per well of input DNA. So we processed all of these samples together through the whole library prep protocol. And then I'm showing you here the fragment analyzer trace for just these no template controls, so just the samples where we didn't put any input DNA in. And we're seeing no library peak, which is good. So this does show that we don't currently have any evidence of any well contamination. Yes, this is a limited data set. We're still building this data up, though these initial results are encouraging. So just to finish up, I wanted to just demonstrate how easy it is to use Firefly to automate time-consuming lab tasks, such as sample normalization. So I just want to give you this example here of how we would go about writing a sample normalization protocol for Firefly. So this is what in this bottom picture here, this is what the protocol would look like on the Firefly user interface. Just 4 steps, 2 liquid handling steps. So what we're doing here is dispensing diluent to an empty plate, so a normalization plate, and then we're transferring in our samples that we want to normalize from our input plate to the sample normalization plate. So this data on the right-hand side is the CSV input. So the data input we can use to determine the volume of buffer diluent that is dispensed to each well of our normalization plate. So the positive displacement dispense head can be used to dispense different volumes to different wells very easily. This CSV, so this CSV input has a very intuitive 8x12 format, which represents our 96 well plate layout. So just by constructing a protocol like this, we can and selecting a CSV with our dispense volumes, we can very quickly and very easily conduct and set up a sample normalization. So these pipetting steps take about 5 minutes to perform. So that concludes what I wanted to take you through today. I hope that I've shown how Firefly is able to achieve repeatable and on-target dispenses from both liquid handling heads. And I hope I've given you an idea of the way in which we work and the care and attention that goes into developing our protocols. In terms of next steps, we're going to continue making protocol improvements and add to the protocols that we're developing. So we'll look at the non-FS NEB kit, DNA kits, and also a directional RNA kit from NEB. We'll also look at the Illumina DNA prep protocol. And once we've looked at those for full volumes, we'll move on to look at these miniaturized protocols for these library preps. Okay, that rounds up the data and information I wanted to share with you today. So thank you for listening.
Thank you very much, Paul and Anita. That was fascinating. We have had a few questions come in during the presentation. For our audience members, you can still submit your questions through the chat box in your attendee control panel. I will call out the questions by the order in which they have been received. Our first question reads, is there an open API available to develop drivers to run with our own scheduling software?
I'll take that one. Yes, is the simple answer. We've got a simple API at the moment that integrators can use. We'd be happy to kind of share those features with us. So if you want to contact us directly about that, we can explore that with you. We'd also be keen to look at the functions in the API that we've already got and any feedback in terms of enhancing that further. So yeah, please contact us and we can discuss that further.
Thank you, Paul. Our next question reads, is the instrument able to integrate to a high throughput process with robotic arm access, for instance?
Okay, I'll take that one, and that leads nicely on from the previous question, because obviously you'd need an API to do that. Yeah, essentially the system as a standalone system is designed to be used as you I didn't really point it out in the earlier bit, but it is enclosed and it has got a door, an interlocked door, so that it's safe. Safe to use in terms of you can't use it with the door open. Also, that prevents any kind of airborne contamination from entering the system. However, the side panels can be removed to give quite an open access area, so for a robotic arm could come in either from the left or the right, or actually potentially even the front as well. But we're thinking more from the left or the right. Typically that would be going into a work cell where then the safety aspect is taken over from the cabinet of the work cell. So simple answer is yes, and it would be and it's been designed in, as I say, with the removable panels. Yeah, okay, thank you.
Thank you, Paul. And here's our next question. Does Firefly support 384 well plate magnetic bleed cleanup?
Do you want to say that one, Anita?
Yeah, sure. So the Firefly will be able to support 384 well magnetic bead cleanup. So we've got all the aspects that you need in order to perform that protocol. We've got the 384 well, the 384 channel air displacement head. We've got the ability to dispense ethanol using the positive displacement head. So we should have no problem doing that. Yeah, we're able to aspirate from our 384 plates on the deck, and we will have a compatible magnet as well. So though we haven't actually investigated this yet, I don't see that there should be any problems doing this on firefly.
Thank you, Anita. One of our audience members says, Anita, thank you for the talk. What was the total time of ultra FS prep on the firefly, including changing the deck with new plasticware as compared to fully manual process?
So it took us about 5 hours to do the process. I think we could have been we could streamline it a little bit further if we're a little bit more efficient with our our user interactions and getting our reagents prepped before before they're required. So in the region of 5 hours, which I think is it's comparable to the manual process, but I mean, usually with the manual process, you're not preparing so many samples. So I think we have the benefit of higher sample throughput.
Maybe just add to that as well that obviously the hands-on, I suppose the thing, and we haven't done it, we should compare the actual hands-on time, because obviously once you then start the run, that's the time you'd normally be operating manual pipettes, whereas obviously the firefly is doing all of that. So I think end to end, it's similar. It's just that there's an awful lot of that time where you're not doing the work the firefly is.
Great, thank you, Paul. Thank you, Anita. The next question reads, does the firefly have the ability to scan barcodes?
I'll take that one. Yes. So initially, we will be offering a handheld scanner, and within the software, there's an option to select scannable racks, or which racks you want to scan or which plates you want to scan. So as you load the system, you scan the racks in. Longer term, we may add an automatic barcode scanner that can do on the fly scanning, but actually in terms of working with beta partners, it seems that way of scanning as you load seems to be quite a kind of popular way of doing it. So that's available as of now, and yeah, we'll determine later whether we add the fully automated scanning option.
Thank you. Here's a great question. Does the firefly replace the mosquito and dragonfly?
I'll take that one as well. No, I mean, obviously it can do to in terms of the dragonfly, obviously it's got most of the dragonfly capability. I say most of, for certain applications like assay development, where there is some quite complex interfacing with design of experiment software, for example, that's kind of on the full dragonfly system, and hasn't yet been incorporated into firefly. The other thing, mosquito is a low volume positive displacement pipetter. Firefly is using air displacement kind of standard pipetting functionality. So in terms of low volume pipetting, so moving samples from A to B, mosquito can go to lower volumes, and it can also manage more challenging liquids. So if it's actual pipetting of difficult samples and low volume samples, then mosquito is a better option. The pipetting of firefly is like most liquid handlers on the market, using air displacement with all the kind of caveats and limitations of that. So you couldn't directly just say it's kind of a mosquito and a dragonfly together. And then it also depends on your workflow, because some workflows may still work better from having kind of modular devices for specific parts of the protocol. So you may find that a workflow may be better kind of with the combination on firefly of pipetting and dispensing together and moving from one to the other seamlessly, or it may be that having a pipetting station, having a separate dispensing station actually works. So we're happy to work with you on your workflows and make recommendations on what would be the best option. But That was a very long answer, but it's kind of yes and no is probably the answer in the end of it. It may be, and it also may not be for the reasons I said.
Sounds great. Thank you. Next question, perhaps this one is for you, Anita. Can you comment on the sample variation you would see across a 96 well plate?
Yeah, sure. So sample variation that originates from, or can originate in different ways. So you can get sample variation if you've got inaccuracies in your pipetting and dispensing. So I don't believe we have issues there with fireflies. So what we can see sometimes is sample variation if we've got time delays between the processing of samples across the plate. So we might get this, and that's because you can't process all samples at exactly the same time. So you might see this more if you're processing samples one column at a time. But the way in which firefly works, we're using the pipetting head by its nature. So all the samples are addressed at the same time, and so we don't really see too much sample variation here in terms of time delays, because they're all processed together. In terms of the dispense head, the samples, or each well is addressed, within several minutes of each other, within only a few minutes of each other. So there aren't likely to be too many sort of time delay, too much of a time delay to show any sample variation. What we can do also is use multiple dispense head syringes to address each, or dispense to each well of a plate more quickly. So that's the way that we can address any sort of time delays there. And what we can do, most of our pipetting or dispense head dispensers are wet dispensers, so to wells which already contain fluid, and so it's not always needed. We don't always get problems with sort of time delays here, because the dispenser is usually followed by some sort of thermal incubation. What we have shown you is that we're able to avoid sort of this time delay between the processing of different samples in different wells. So for instance, the way we dispense our ethanol to our plate, we just do a low volume dispense, say 20 microliters to each well of the plate, only takes a minute. This prevents our beads drying out, and is a way of getting around any sort of time variation you might see across the plate. So whilst we haven't got data for a full plate of samples yet, we're working on that. I believe that with firefly, the sample variation shouldn't be much more than what I've shown you here today.
Thank you, Anita. Well, let's see. I think we can squeeze in one more question. It says, how reusable are the tips for the 96 well head tips? Have you used a washing through or cleaning tray?
I'll take that one. So in terms of, so let me start with the reusable, because there's two aspects to it. Firstly is the sealing of the tips, because most liquid handling systems use a kind of forced on tip, so it's kind of a deformation seal in terms of the tip mandrel is forced into a tip, and then when you eject it, if you pick it up again each time you put in force, and that does tend to wear the tip and cause issues over time. So that tends to be a finite lifetime, and that you hear often people talk about, 5 to 10 reloadings of the tips, but even after one reloading, you may start to see performance variations on the tips, which is obviously a concern. The way that the tip loading system works on the Firefly is that the tips are in clusters with a collar, and it's actually a gasket seal against the collar. So there is no deformation of the tip, and actually you can reload that as many times as you like, and there's no, it's very low force, and there's no kind of change to the tip seal that you see. So in that respect, yes, you can. You can kind of just keep using a set of tips. So within a protocol, if you're happy at certain stages to revisit the same set of tips, you can do that, and obviously save your tip consumption that way. There are also now tip washing systems on the market that we've been talking to one company recently that's doing that, and they've already validated the Firefly tips on that system. So you could employ something like a tip washing system. The pipetting head on the Firefly is common to the apricot systems that we also supply, and on some of those systems they do employ tip wash stations where, you know, perhaps where people are a bit less sensitive to contamination. We haven't looked at that on Firefly yet, but there's no reason why a tip wash station couldn't be added at some point as well. So so certainly reusing tips from the sealing point of view is no problem. Depending on the application, tip washing may be an option. And then, you know, if you're looking at some of the tip washing systems offline and you're happy to use those, then again, that the tips are compatible with that type of system. Yeah.
Thank you. Thank you for that, Paul. Thank you for all those questions, and thank you again, Paul and Anita. Apologies that we haven't managed to answer all your questions today. However, our experts will endeavor to address them individually in the coming days. Also, please feel free to get in touch to send us any other questions about today's presentation. You can email us at marketing@sptlabtech.com. On behalf of SPT Labtech and our presenters, thank you for joining us and enjoy the rest of your day.